The goal of the project is to try to enhance, the growth of the beneficial bacteria. So the process that Jay was describing is going to be optimized. It's going to be faster, and then we can, basically eliminate the ones that won't be beneficial. So some of the bacteria that we isolated here from the rocket composter, they grow at 55 degrees. So they don't grow at 35. They don't really like any other temperature. So that's what we call thermophilic bacteria. So we're trying to develop those bacteria so we can hopefully digest, paper or any other material using high temperatures. There are other types of bacteria that we found that they like a specific media, low pH. So we're finding out different species, so hopefully down the road we can develop different, like procedures and treatments to basically target different types of waste.
It's actually fascinating. So it's called the Rocket. It's a big compository that takes all the trash from the cafeteria and it turns it into soil, which I think is cool. And we take samples from the soil. We take some from the top and some from deep down inside to get different types of bacteria. We come back here. We do a couple stains. We do gram stains. We do a lot of things. We do diluting process. Basically trying to figure out what bacteria is inside the soil. So we come through here. We do the stains. We come over here. We count the colonies. We see which ones are different. We take samples. We do gram stains to figure out what exactly and who they are. Then we can go over here to this machine over here. This is to extract the DNA, and this, this is to help us better figure out who exactly is the bacteria that we're doing research on.
So that's a thermocycler. It's used, to amplify the DNA. So we're looking for different types of bacteria, and we have a specific DNA fragments that are going to be amplifying different types of, microorganisms.
And then with this, we actually just open this up. We put little tiny test tubes with the DNA we extracted from the colonies. We put it in, it incubates for over like minimum 6 hours, maybe more. We get the idea of who they are. We also have another cool project that I wasn't here for the beginning of this, but the professor and another colleague of mine, Ed Eduardo, they actually found soil from back there and they put it into these little containers and they sealed it up. And the soil, the bacteria inside the soil was actually emitting electricity. So we were trying to figure out who the bacteria was that was actually emitting the electricity. So we did some tests. After a while it did die off because they opened to try to transfer the bacteria over. It did die. But amazingly, after a few months of no activity, it came back by itself. We didn't do anything, it just came back. So I found that extremely fascinating.
Okay, so what I'm doing now is I'm taking the slide that I took from the rockets that we have upstairs, and I'm trying to check the morphology on it to see if it's a rod or a cockeye, which is the beginning of the identification process. From what I can see now is that this bacteria is a gram-negative, so I'm just trying to check now to see if it's a rod. You can't see it at the lower magnification, which is when you put it up to maybe 100 or 40. And due to the fact that I changed it to 100 now, I'm going to change the condenser and the fine adjustment to make sure that I can see everything perfectly. And now I have a perfect picture, and it is a gram-negative rod.